Hi everyone, welcome back to Teacher Kayal's Guide to Biology A+. In this video, I'll guide you through answering SPM trial questions on the topic movement of substances across a plasma membrane, focusing on structure of plasma membrane and transport across the plasma membrane. A phospholipid has a head and two tails. The head is polar, which means it has electric charges. The electric charges attract water, so the head is called hydrophilic. Hydro means water, philic means love. So hydrophilic means water-loving. The tails are non-polar, which means they have no electric charge. Because they have no electric charge, they do not attract water. So they are called hydrophobic. Hydro means water. Phobic means fear. So hydrophobic means water fear. These characteristics cause the heads face water, while the tails hide from water. Glycoproteins are receptors for hormones, which means that when a hormone binds to a glycoprotein, the glycoprotein will cause a response inside the cell. Glycoproteins also act as antigens on cells to differentiate between cells that belong to the body and foreign cells that do not belong to the body. This identification allows the immune system to protect the body's own cells and destroy foreign cells, such as pathogens. In addition, glycoproteins also help to stabilize the plasma membrane by forming hydrogen bonds with water. For a 2-mark question, write the top 2 points. For a 3-mark question, you need to write the extra point. Cholesterol in the plasma membrane stabilize and strengthen the plasma membrane. This makes the plasma membrane less permeable to water-soluble substances such as ions and more flexible. For a 2-mark question, write the top 2 points. For a 3-mark question, you need to write the extra point. If the plasma membrane contains less cholesterol, the plasma membrane will lose stability, causing the plasma membrane to become more permeable to water and ions. If there is no cholesterol, the plasma membrane becomes too fluid. Too fluid means the phospholipids move too freely, causing the plasma membrane to become too permeable to water and ions. In extreme cases, the plasma membrane may rupture. Scientists make use the distinguished feature of the selectively permeable membrane by creating artificial phospholipid bilayer called liposome to safely deliver drugs to precise destination. First, the drugs are packed inside the liposome. To ensure it reaches the correct target, scientists attach specific molecules, such as glycoprotein molecule, onto the surface of the liposome so it can only bind to receptors of target cells. During transport in the body, the liposome protects the drugs by preventing unwanted substances such as acidic gastric juice and enzymes from entering and damaging the drugs. Therefore, the drugs remain safe inside the liposome. Only once the liposome binds to the matching receptors on the target cell surface, the liposome releases the drugs. The way liposome delivers drugs safely and precisely to target cells is very similar to how liposome delivers vitamin C. If you feel the way liposome function sounds so complicated for you to understand, you can simply imagine a liposome as a waterproof package that protects its content as much as possible from the external environment when transporting it to a precisely stated address on its cover. The drug or vitamin inside the liposome is like the item inside the package, and the phospholipid bilayer is like the waterproof covering that prevents damage during transport. The targeting molecules on the liposome surface are like the delivery address written on the cover of the package, ensuring it reaches the correct destination. In contrast, traditional drug and vitamin delivery methods without liposome are like a non-protective package where water can enter and damage the content as well as the cover of the package. Lipid-soluble molecules such as fatty acids, glycerol, vitamin A, vitamin D, vitamin E, and vitamin K can move across the phospholipid bilayer. Lipid-soluble molecules such as fatty acids, glycerol, and oxygen can move across the phospholipid bilayer because they are hydrophobic. As mentioned earlier, lipid-soluble molecules such as oxygen can move across the phospholipid bilayer because they are hydrophobic. They move by simple diffusion down the concentration gradient. When you are asked to draw a channel protein, remember to include a pore. Ensure the size of the channel protein is proportional to the other parts of the diagram. Lipid-insoluble molecules such as glucose, amino acids, and ions cannot move across the phospholipid bilayer because they are hydrophilic. In addition, glucose and amino acids are large molecules. They move by facilitated diffusion through carrier proteins down the concentration gradient. The diagram and the table show the differences between transport across the phospholipid bilayer and transport through carrier proteins. Carrier proteins can function in both passive and active transport. However, the carrier protein shown in the diagram is not using energy and moves molecules down the concentration gradient. Therefore, it is carrying out facilitated diffusion. Water is not stated under the phospholipid bilayer because only a small amount of water moves slowly through the phospholipid bilayer. Water mainly moves fast through channel protein. All active transport occurs through carrier proteins. An example of carrier protein is sodium-potassium pump. First, 3 sodium ions from cytoplasm bind with the carrier protein. Then, ATP provides energy for the carrier protein to change shape to face the opposite side of membrane. As a result, 3 sodium ions are transported out of the cell. Based on the diagram, substance P is being transported into the cell by active transport through carrier proteins using energy provided by ATP against the concentration gradient. Both passive transport and active transport involves the movement of substances across the plasma membrane. Both processes occur through a selectively permeable membrane. In addition, both passive transport and active transport control what enters and leaves the cell to maintain the cell's internal conditions at the same level so the cell can function normally. Both processes also involve the kinetic energy of molecules. This means molecules are always moving randomly because they have kinetic energy. In passive transport, this allows substances to naturally move from a region of higher concentration to a region of lower concentration. In active transport, molecules also have kinetic energy, but the cell uses additional energy from ATP to force substances to move against the concentration gradient. Passive transport does not require energy, while active transport requires energy from ATP to change the shape of carrier proteins. Passive transport occurs down the concentration gradient, whereas active transport occurs against the concentration gradient. Passive transport continues until dynamic equilibrium is reached, meaning the concentration becomes equal. In contrast, active transport allows cells to accumulate needed substances or remove unwanted substances, mainly mineral ions. Passive transport can occur with or without a selectively permeable membrane, whereas active transport only occurs across a selectively permeable membrane. That's all for this video, and I'll see you in the next video. Remember to study smarter, not harder.